Thapa from class 12, and today I am excited to talk to you about DNA and the fascinating process of DNA replication. Why is it not connected? See, this is not connected. It's fine. It's fine. So the key topics of today is what is DNA, who discovered it, and why is DNA important, and the steps of DNA replication. Now let's start off with what is DNA. DNA or deoxyribonucleic acid is the genetic molecule that carries the genetic instructions for life. Think of it as the blueprint that determines how an organism develops, functions and reproduces. Now DNA is made up of two long strands that form a double helix structure. Now, who discovered this double helix structure? This double helix structure was first studied by Franklin and Wilson by using X-ray crystallography, which laid the foundation for the later study continued by Watson and Crick in 1953. Now, as stated before, DNA is made up of two long strands that form a double helix structure. In this, each strand has a 5' end and a 3' end and is composed of smaller units called nucleotides. Each nucleotide has a sugar molecule, a phosphate group and a nitrogenous base. Now as you all can see, we had a 5' end and a 3' end. That's great. Next up we have 4 nitrogenous bases of DNA. First is A, adenine, then T, thymine, C, cytosine and G, guanine, of which your A and G are purines and C and T are pyrimidines, in which A forms a double hydrogen bond with T and C forms a triple hydrogen bond with G. And the two strands are anti-parallel to each other and both run in opposite directions. Now let's move on to why is DNA important. DNA is crucial because it contains the information needed for an organism to grow, develop and maintain itself. It contains the genetic information that is passed on from one generation to another, ensuring the continuity of life. Now that you all have a basic understanding of DNA, let's move on to our main topic, that is DNA replication. DNA replication is the process by which DNA makes a copy of itself. Now for you all to understand it much more better, we will show you DNA replication through a role play. But before we go into the steps of DNA replication, let me introduce you to our important enzymes of DNA replication. First up we have a DNA helicase, which helps in the unwinding of the double helical structure. Next up we have our RNA primase, which synthesizes short strands of RNA called RNA primer. Third, we have our DNA dependent DNA polymerase, which adds nucleotides to the template strand. Next, we have our DNA ligase, which seals the gap between the Okazaki fragments on the lagging strand, helping to create the new strand of DNA. Now, as you all have met the important enzymes of DNA replication, let's move on to the steps of DNA replication. First step is initiation. In this, DNA replication begins with the unwinding of the double helical structure. The unwinding of the double helical structure is done by an enzyme called DNA helicase. Now, helicase is responsible for breaking the hydrogen bonds between the nitrogen bases, which creates a replication fork. And the two separated strands then acts as a template for creating a new strand of DNA. So now you can see we have two separate strands and a replication fork was created, which will now act as a template for creating a new strand of DNA. Now let's move on to our second step, that is primer binding. In this, an enzyme called RNA primase synthesizes short strands of RNA called RNA primer. As your DNA polymerase can only add nucleotides in one direction, that is 5' to 3' direction. Now our RNA primase synthesized short strand of RNA called RNA primer, which created a starting point for DNA replication. Next, the third step is elongation. In this, our DNA polymerase adds nucleotides in the 3' end of the RNA primer, extending the new strand of DNA. Now in this we have our two strands, that is the leading strand and the lagging strand. First up, let's see our leading strand. Our leading strand is synthesized continuously and in one direction, that is from 5' to the 3' direction. Now you will see that our DNA polymerase adds nucleotides in one direction, that is from 5' to the 3' direction. One base is added, then the second one is added, and then the third continues, and then we have our last base. So now our leading strand is completed. Let's move on to our lagging strand. Our lagging strand cannot be synthesized in a continuous way as it runs in the opposite direction. So now our DNA polymerase adds nitrogen bases in a discontinuous way, and each DNA fragment starts with a short RNA primer, and then the DNA polymerase adds nucleotides in the 5' to the 3' direction. And then further down the line, a primer is added and another Okazaki fragment is created. The lagging strand is synthesized in short segments called Okazaki fragments. Why? Because it is discontinuous and it cannot go in a continuous way. See, now our lagging strand is completed. Now let's move on to the next step of DNA replication, that is primer removal and replacement. After the new strand of DNA is formed, the RNA primase is removed by the DNA polymerase. And then this enzyme fills in the gap with DNA nucleotides. Okay, so next up we have our fifth step, that is ligation. After the new strand is formed, we have an important enzyme's role here, that is DNA ligase. Your DNA ligase will seal the gap between the Okazaki fragments and create a new strand of DNA. It seals the gap between the Okazaki fragments on the lagging strand, which creates a new strand of DNA. So a new strand of DNA is created. Now the last step of DNA replication is termination. That is, this DNA replication continues until the entire molecule is replicated. Now some key points to remember from my presentation is that DNA replication is a semi-conservative process. That means when a newly synthesized strand is formed, it consists of one old parental strand and one newly synthesized strand. And the accuracy in DNA replication is very crucial. And your DNA polymerases have proofreading abilities to correct the errors. In conclusion, DNA replication is an essential process for accurate transmission of genetic information during cell division. Thank you all for your attention, and I am happy to answer any questions you may have.
I didn't quite get the part where they dance together, like what did that symbolize?
Oh, that was DNA polymerase going on with RNA primase, because that's how it starts, right? Primer binding.
Also, the yellow tag thingy with the red dot.
Yeah, they were removing the RNA primer. As I said, no, the enzyme DNA polymerase also fills in the gap with the DNA nucleotides. So that's why it's removing the RNA primer, because that step, the fourth step, right? It is RNA removal and replacement. Okay? Yeah.
once the DNA strands are separated during DNA replication, there is a key element that does not allow them to bind back. Do you know what is the key element?
No, I'm so sorry, I don't know.
What phase of the cell cycle exactly does all of this happen?
The S phase, that is your synthesis phase. Okay? Thank you so much, everyone.